everyone. So welcome to another session of What Can I Do with My PhD seminar series. It's my pleasure today to introduce our speaker, Karen Colwill. I hope I pronounced your name right.
Yep.
So Karen is the Director of Operations of the Network Biology Collaborative Center at Sinai Health in Toronto. She has a PhD in molecular genetics from U of T, as well as completed her postdoc there as well. For the past 20 years, she's been working at the Lundfeld Tenenbaum Research Institute in various different roles, anywhere from research associate to a project coordinator, and now the director of operations. And today she's going to discuss with us her journey and experiences in academia in a non-PI senior role. So welcome, Karen.
So yeah, thank you for inviting me. So what I'm going to do today is kind of tell you about my route from my PhD to where I am right now as the Network Biology Collaborative Center Director of Operations. And what I'm showing you with this screen is kind of the route that I took to get to where I am now, starting with my PhD and with a brief stint in industry before I circled back into academia and into the same institute where I did my PhD to become a research associate, all the way through to when I became my current role as a director of operations for a technology center. And there's some of my slides are a bit text heavy, so please bear with me through them. So start this talking about my PhD, which I did at the University of Toronto. It was a PhD in molecular medical genetics with my supervisor, Tony Pawson at Mount Sinai Hospital. And I did this from 1991 to 1996. And during my PhD, I identified a role for the click kinase in regulating a group of SR splicing factors. And when I finished my PhD, I knew I wanted to continue in research, but I wasn't sure where I wanted to do it. And at the time, I was married and my husband was working in Toronto. And this is a bit of a common theme, particularly through my earlier career decisions, which was, how to combine my love of research with some of, my work, my life. So because my husband's working in Toronto and I didn't want to disrupt his career, I did choose to do a postdoc, but I did it in Toronto and I did it in the lab of Dr. Brenda Andrews. And I did it from 1996 to 1999. And during my postdoc, I characterized the domain structure and functions of a cytoskeletal protein. It was also during this time that I had a 6-month maternity leave. And when I was in my postdoc, I realized that I still wanted to continue in research, but I didn't want to go the route of becoming a principal investigator. And this is a personal choice on my end. I was interested in science, but I didn't feel like I was passionate enough about it that I wanted to dedicate so much time and mental effort to it at the time when I already had one child and was hoping to expand my family. So what I decided to do was to take a job in industry. And the job was at a new startup company called MDS Proteomics. And the goal of this company was to identify new therapeutic targets and eventually to develop new therapeutics by using mass spectrometry to identify protein-protein interactions. And this was really at the start of when mass spectrometry was being applied in this way. And the way the company was doing this was sort of setting up almost like a factory-like way of processing samples, where they would identify proteins of interest, clone them, express tagged versions of them in cells, enrich the tagged proteins and any other interacting proteins, and identify those proteins by mass spectrometry. And of course, there would be data analysis involved in this. And my role was really in overseeing the cell biology and protein enrichment areas, where I had to, we were a young company and everything was just starting up. So we had to recruit, train, and manage staff. We developed standard operating procedures, including quality control. There's assays that had to be scheduled across this whole continuum. data to be reviewed, troubleshooting of all the data, of course, interaction between all the team leads and clients who were interested in using our services. I also became the manager representative for health and safety. So a lot of times when you're in these roles, you have non-scientific roles as well. And what was important to me at the time was, by moving into industry, and it was at a time when maternity leaves were extending, that I was able to have a 1-year maternity leave. But unfortunately, by the time I came back from that 1-year maternity leave, the new startup company was running out of money, and they no longer had a position for me. So at that time, I was looking for a job, and I'd been out of science for a year at that point, and I had 2 young children. So all these considerations of my children, influenced my decision for my next step. And at the time when I was thinking about what to do, I was actually contacted by my PhD supervisor, Tony Pawson, because he was looking for someone to help him out as a bench level researcher. And he hired me on a 6-month contract. And shortly after that, he offered me a full-time position. What I was doing for him will look very similar to what I was doing at MDS Proteomics, because again, it was a new time for this application of mass spectrometry to identify protein-protein interactions. And I already had experience running a pipeline, and I did essentially the same thing for him, although I expanded it out to assisting with the cloning. And I was working with trainees in his lab, postdocs and PhD students who wanted to run large-scale projects and needed some technical assistance along the way. And I was also still performing validation experiments. And after I was doing this for a while, I talked to Tony and said that I wanted to do more of a coordinated role and move away from bench work. And part of this was, I was good at bench work, but I thought that I was much better when I was in a role as a coordinator. And it was also more compatible with work-life balance. Working at the bench is great, but sometimes experiments can take longer than you plan. And then when you have to go get a child out of daycare, etc., it gets challenging. So I asked if I could do stuff more on the coordinating role. And he agreed. And one of the first things I did was work on an application to Genome Canada for a grant. It's a multi-year, multi-PI, multi-institute grant. And in the end, we were successful. And it's probably the largest grant I've ever worked on, would be my first one. And we received 13 million, 13.7 million in funds direct from Genome Canada. And they asked that you match those funds with other co-funds. So the whole project was worth 27.5 million. So here's my role. And I just put a little icon here to be more on the coordination side. So with these large grants, there's a lot of coordination in actually getting the grant written, and I was in charge of that. It went across multi-PIs, multi-institutes. I helped create the budget and all the justification. When you write one of these grants, there could be 100 pages and only 20 pages is directly linked to the science. The other 80 pages, you have to write about how you manage the grant, what the benefits are to Ontario or to Canada. What is the justification for what you're asking for in the budget? These tend to be what they call milestone-driven grants. So you say that you commit to doing specific tasks at certain points, and we had to create those. And these grants, you have to go to face-to-face meetings with the scientific review committee, and they'll ask you questions and you have to defend it. My role was to defend all the non-science parts of it. When you get the grant, you have to organize, as a coordinator, you have to organize team meetings. There's financial and scientific reporting. For Genome Canada, it was quarterly. We had to fill out these reports. Often they're just annual. We had to explain if we hadn't met our milestones, why not, and what we would do to make sure that we completed our deliverables for the project. And this particular grant had a due diligence review partway through, so we had to write a full report on our progress and again go and defend it. And then when the grant closes, you have to write final reports. And even before that, you built up all this infrastructure, senior people, including myself, that if you want to continue, you would not need to find new funding for. So you always had to consider what are the next new grant applications out there. And while I was doing this, and this is a common theme for me, is I always wanted to kind of keep in a scientific role as well. So while I was doing all the coordination, I was also overseeing some of the science goals for the Pawson lab. And I was looking and coordinating the proteomics, mass spec, and computational teams that were in Tony's lab. And we also had within the Institute all the mouse and human clones. And for the Institute, so I worked with a program to create an information management system to track all the reagents for the Institute. I should say that, like, I actually, it was the having gone through the process of applying for the grant and getting it awarded that led to my promotion from a research associate to a project manager. And that's often the case that you're in your role and taking on new responsibilities, and then, you know, you're recognized for that by a promotion. The next promotion I got was to a staff scientist. The staff scientist performs a senior role in an institute, but they're not a PI. But they're not providing support just to one principal investigator, but they're providing support to the institute as a whole. And at least at Sinai, these were 3-year renewable terms contracts, and the salary is guaranteed by the institute. And this is different than the role I had as a research associate or a project manager, where, you know, the PI is responsible for making sure that they have the funds to pay you. Here, your salary is guaranteed by the institute. Of course, it could be offset by grants, but if there are not grants, the institute commits to paying the salary. And the appointment has to be, and the renewals have to be approved by the Institute Executive Committee. So the reason I got a staff scientist position was for the role I had in coordinating these multi-PI grants that went across the whole institute and even beyond to other institutes. And it was for large grants. I mentioned the one for Genome Canada. Ontario also has grants that require project managers. And we're also put together a lot of grants for new infrastructure for our facilities from the CFI, and I helped with those. I was also at this point helping to coordinate samples that came into the proteomics facility, not just for the Pawson lab, but for the Institute. I mentioned the open prelims that we had, and at this time, I was still contributing to scientific research. I was also during this time that Tony Pawson, who I was working for, passed away. I don't know how many people know Tony Pawson. Back before I joined his lab for my PhD, he discovered the SH2 domain. And he was really instrumental in defining a role for non-catalytic domains to have a big contribution in signaling. And he did a huge amount of work mapping signaling downstream of tyrosine kinases. And he was recognized for this by winning the Kyoto Prize, the Gardner Prize. He became a companion of honor for the Queen. So this happened during this time, and it was shortly after this that I took on a new role as the Network Biology Collaborative Center Manager. This wasn't an official title. I was still a staff scientist, but I was a manager from 2014 to 2022. And the Network Biology Center was established in 2014. And what we did is we amalgamated several facilities within the Institute into one center, with the idea that the center would provide service to Canadian scientists in functional genomics and proteomics. And for the past 8 years, it was one of 10 technology platforms across Canada that was funded by Genome Canada. And my role in this was to manage grants linked to the center. The Genome Canada was the main one, but also we had funding from CFI and the Terry Fox Research Institute. It was important to the center to budget, forecast, review financials. There was it's a cost recovery center, so there was billing and payments, communications to let people know what the facility does, our social media, meeting with clients, always recruiting staff, and helping out as needed. Just want to give you a brief idea of what the NBCC is. It's managed by 2 scientific directors who are senior scientists at the Lunenfeld, Jeff Rana and Anne-Claude Gingras. And I've just given you the website. You can follow us on Twitter if you want to find out more. And it's comprised of 5 facilities. What I'm doing here is I'm just showing you that each of these facilities is managed by a senior level scientist, some at PhD level and others that have up to like 15 to 20 years experience working and managing facilities. The proteomics facility is mass spectrometry based, involved in protein identification and quantification. And it's really focused on protein-protein interactions or mapping proteins in close proximity to each other. We have a sequencing facility, and our specialty is in bulk and single-cell RNA-seq. And we do a lot of barcode reading for CRISPR screens. We have a high-throughput screening facility that has 2 automated platforms. And it's used predominantly for drug screens, and as I'll show you, for ELISAs. And we can do assays in vitro using like purified proteins. We can do them with cell lines, and we can now do them with 3D tissues and organoids. We have a high content screening facility, which is really automated imaging. It works a lot in tandem with the automated facility. So as an endpoint readout could be looking at cell morphology, expression of a fluorescent protein or a translocation of a protein. And then we have high to super resolution imaging, which is really when you really want that fine, super detailed imaging. And then this can be done over time. And the idea of putting all these facilities together was for really better coordination for the clients, because you can imagine that a client may get a new hit in a screen, a new protein that's important in a pathway, but they don't know what it does. So they could, do some mass spec on it. They could do some detailed imaging of it. So it's this kind of idea of helping clients go across all these technologies, which is one of the reasons the center was created. So in 2022, based on the work I was doing, I was promoted to become the director of operations for the NBCC. And as a director, it's just really a bit of a more higher level up, forward looking than a manager. So you're really making sure that the facility managers that you support have the resources they need, and you provide direction. You look at how you can improve cost savings and efficiencies across the center. A big part of what I still do is work with the directors, on new funding, including grant writing. Think about what the long-term objectives we want for the center, the budgets for it, financial forecasts to make sure the center stays sustainable. Still a lot of work on interfacing with grant agencies and external institutions. And again, keeping that scientific hat, coordinating projects and clients that span those different facilities, and lead large-scale projects, help with manuscript writing and review. So what I'm going to do, having kind of explained my career path and where it's gotten me, is just kind of show you how this could be all put together and how we helped out over the last 3 years in the COVID-19 pandemic. So as soon as the lockdown happened in March, we were all, as everyone was thinking of how we could help out. What we knew is that the NBCC had the technology and expertise to rapidly pivot to COVID-19 research. So on our mass spec proteomics facility, we identified proximity interactors for SARS-CoV-2 proteins. We adapted an automated barcode screening strategy to sequence variants of concern. And then we could track the rise and fall of some variants, and then of course the rise and continuation of the Omicron variant. And we developed assays to help with COVID-19 serology. And right now we've contributed to over 40 publications, and that number is still climbing. So I was really involved in building out assays to measure antibody levels. We did this by an ELISA-based assay. And I'm just showing a schematic of the spike protein binding to the target host cell. We have the full-length spike trimer, its receptor binding domain, and the nucleocapsid. And in the ELISA assay, we take one of these antigens and absorb it to the bottom of a plate. We add primary antibodies that come from patient serum or plasma, or they could be purified antibodies. Add a secondary antibody linked to an enzyme MHRP, when its substrate is added, it emits light or color. And just 2 weeks after the lockdown, we had our first result, and Bhavisha, our technician, lab manager, Bhavisha, and PhD student Kento were the ones that did this initial work with samples and reagents from U of T. And you can see that when we added samples from someone who had been positive for COVID-19, we got a strong signal, but not from those prior to COVID-19. So having shown we could do the assay, our next step was to build up the automated assay so we could go through thousands of samples using our high throughput screening facility. And again, just thinking about the logistics of this, which was my first role in this, is we had to work with our biosafety officer to get approval for running the samples. This is very early on in the pandemic, and we weren't really sure what the safety risk was in bringing these samples in-house. Procuring samples during the pandemic, not an easy task. Supply chains were interrupted, and there was a lot of demand out there for plasticware. Like pharma companies, particularly even after vaccination, they were using up some of the plasticware as fast as they could, and we were also trying to get it. We needed to get samples to test within the Toronto community, and we worked with the National Microlab, and all this involved research ethic board applications, material transfer agreements. We needed funding. We had to apply for grants. We worked with our foundation and our finance department, and we had to bring people on to help out. And you can see that just from this, how many, in a role as a coordinator, how many different departments and experts that you need to work with, both in your own institute and across institutes. And through this, I was also involved in assay development. We were working with the high throughput team to adapt the manual assay to automated. We needed to identify controls, figure out what dilutions to use, make sure we had the right reagents, develop SOPs, how we were going to analyze the data. And we had to set up ways to track both the samples and the reagents. So I'm just going to show you a little bit of data now. This is the first version of our assay that we published in the summer of 2020, going into the fall. And at this time, remember, none of us really knew if you were infected, how long, the immunity may last. And we were using antibody levels as a proxy for this. And some of the early reports using different assays were suggesting that antibody levels were really transient. And what we showed here, and other groups also at the same time were starting to show this, is that if you look at antibodies to spike or its receptor binding domain, going out 115 days, and this is a relative ratio, which is just the raw signal we get normalized to a recombinant antibody standard. This dotted line is the threshold for positivity. So you could see that negative, someone who no COVID-19 were negative. And you could see that people would mount an antibody response starting after about 15 days. And then going at least 115 days, they were still positive. And this was important in reassuring that there was some immunity that we could get from, at this time, infection and later on from vaccination. We continued to develop this assay, and we published a version 2.0 that I was the lead author on in 2022. And by this time, we'd worked with the National Research Council of Canada to get highly pure, large batch reagents for it. We were working with a partner lab in Ottawa that was also performing the same assay. And we calibrated our assay to the WHO international standard. So what I'm showing here for all of our 3 antigens is the results from a study the WHO did on 5 different samples, negative up to high levels of antibodies. And these are the aggregate results from about 37 different laboratories. And this is just showing that both us and the lab from Ottawa were well within the range of what others across the world were seeing, validating our use of our assay. So as we were building out these assays, and Claude in particular, who was the PI lead for this, and I as well, were working with the immunity task force that Canada established for COVID-19, working on in their task force And one of the things they did is they sent out samples to a lot of different labs to see, for cross-validation, to see who it makes sense to do some of the analysis. And these initial samples, it turned out that our lab and our partner lab in Ottawa, had really good results and were able to do it high throughput. So the Immunity Task Force made us a reference lab for their studies. And as a result, we've now analyzed over 200,000 unique samples. So starting again with logistics that I was involved in, this now meant that I was coordinating over 30 different projects. We had to prioritize samples to help inform public health decisions. This is particularly after vaccination. We were getting samples in from long-term care home residents. Were they responding? How soon should we boost them if their antibody levels were dropping? We were getting samples in from vulnerable populations, such as those on dialysis. One of the things we showed is that people on dialysis, antibody levels were dropping, and that helped prioritize them for booster doses. We were also kind of informing what vaccine was working the best in older people, long-term care home, etc. The Moderna vaccine was leading to higher antibody levels. So it's important, as all these samples were coming in, to figure out what samples needed to be run first. Sample procurement always continues to be an issue, and paperwork continued on, and more so as we were dealing with more grants. We kept expanding, so we had to keep hiring people. And we needed new equipment, and some of our equipment needed replacing. So this was always involved in keeping the things to keep this assay running. On the scientific coordination side, we had to meet with teams. We were helping them design their assays. We were providing results and explaining results back to them. All the data that we had needed to be reviewed. I was working with data specialists to develop database and what reports we would send out. There's issues with any of the running that we had to troubleshoot, updating procedures, always thinking about writing grants and manuscripts. And our assay had to change with the pandemic. When vaccination came, it shot the levels for a spike in RBD so high, we had to add extra dilutions. At the beginning, when we were just measuring who in the population had the disease, we had to be very careful about false positives. But as more and more people got the disease, we were just as worried about false negative. And this meant we had to review our thresholds and figure out if they still made sense. Variants of concern kept arising. We were doing, I'm not talking about them, but we were doing neutralizing assays. And every time a new variant of concern came, we had to add that into a repertoire of things to test. Just going to give you just a couple of data slides, just to kind of get a feeling for what the data looks like and how it helped. One of the projects we had was a CanPath project, and this is a longitudinal study across Canada involving hundreds of thousands of Canadians. And we worked with them, and they sent us dried blood spots from 20,000 different individuals. And the timing of this was right around the time of vaccination. And they were able to record, this is looking at receptor binding domain and spike, the relative ratio, which is measuring the antibody levels, time since second vaccine. And you can see this is a seropositivity threshold. So this is reassuring that you could be up almost a year from your second vaccine and we could still measure antibody levels. And one of the things we're looking at for vaccination was vulnerable populations. I mentioned long-term care home residents, but there was also a large component of elderly people living in the community. And the Stop COVID study, which was completely online, signed people up online, sent them out dried blood spot kits, was looking at how community living elderly compared to younger cohort. So they recruited people 30 to 50 years and then 70 plus, and they tracked them from their first vaccine dose. And this is looking at the receptor binding domain as spike. And you can look, as soon as they got their first vaccine, all of them passed the seroconversion threshold. But the younger folks in the study had higher antibody levels. And prior to the second dose, those that were elder, actually the median level was below our seroconversion threshold for positivity. And getting that second boost, you can see increase the levels for both cohorts. Again, a bit more for the younger threshold. And it boosted them above the level that we see for people who are infected. And you can see that over time, you get a decrease in antibody levels that get increased with booster doses. And what was interesting in the study is 48 weeks out, they could see that now the 70 plus have the higher antibody levels. And this is because, you know, the elderly people, knowing they were more vulnerable to COVID-19, were going out and getting more booster shots. And you can see that for them, that paid off, and their antibody levels were high. And this is very reassuring to know that we could protect those vulnerable in our population by giving them booster doses. And although I did say protect, I just want to qualify, we don't know what a correlate of protection is, but we do know that the higher antibody levels you have, the better you are to fight off an infection and reduce its severity. So I just want to end here on just a little overview of what I think the key things for my success were. For me, it's always been important to be in a positive work environment, to have people that I work for that are very supportive of me. Over my career, I've tried to ask for things I want. And as an example, when I was on the contract with Pony, and he asked if I wanted to work full time, it was very important to me. I was a young mother. I wanted to spend as much as I could time with my kids. I asked if I could have like the ability to work from home sometimes when it made sense. And I asked if I could just take a day off here and there just to be a mom. And he agreed to those things. And that was important to me. I love research. And I like to coordinate and manage it. And I never wanted to go into a position where I would only be doing project coordination or management. I really wanted to maintain both. And I was lucky and fortunate to be able to do that. Through the talk, you can see that for me, work-life balance was important, and that being able to manage those and as much as anyone can manage them was important to me. Science is a very small world. I mean, you could see right in my first slide, I circled back after I lost the position in industry to my PhD lab and continued my career from there. So I just, you know, it's very important to always kind of work hard. I think it's important to be generous with helping others. I was always offering my help, willing to do anything to get things done. And when you're working with like a finance and other things where they have a lot of deadlines, being able to say, here, I can help with that, I think made a big difference in how I succeeded. Lastly, I just want to kind of thank some of the people I had, you know, Tony Pawson as my supervisor, Brenda Andrews. When I came back to the Lunafelt, I've been working with their directors of research. The NBCC has its own directors, including Laurence Pelletier, who oversees the imaging part. I have a wonderful team on the serology I talked about, and all the managers. And I can stop there.
Yeah, thank you so much for sharing your experience with us. I know we had a question, so if anybody's interested in asking a question, you can put it in the chat, or you can even like raise your hands using one of those emojis and ask the question directly. We did have a question from Leslie asking, you talked about the importance of a positive and supportive work environment. How do you identify these before you really accept the job? Yeah.
For me, it was a bit easier because as I went from spot to spot, I did end up working. Like, for instance, when I did my postdoc with Brenda Andrews, she was on my committee as my PhD. The next spot I went was a startup company out of Sinai. So I'm probably not the best person to do it, but I think if you are in a position and you're applying for a job, and if you can, don't just talk to the person. See if you have a chance to interact with the people in the lab and just ask them, you know, how they find the work environment and what, you know, they think would be important to succeed. I know when we interview people, we always bring them in to meet the members of the lab. And that gives anyone who's interviewing the chance to kind of know the culture of what they're getting into, because that's important. I think it's not only can you do the work, but can you interact with your colleagues? Can you talk to your boss? Are you happy to go to work and stay motivated? And I want to have a good mentors and boss, but I also want to have the people that work for me have that experience as well. So, I think it's very important when you're looking to see if you have that chance to interact and talk.
Right. So going
colleagues.
off of do you do a lot of hiring in your position? And like, is it usually like grad students you're hiring or staff or technicians?
Yeah, so I'm not a PI. I work very closely with PIs like Anne-Claude Gingras and Jeff Rana, but they would, they're the ones bringing in the trainees into their labs. And, sometimes as they're interviewing them, sometimes they come through the lab and they talk to different people and we give input into it, but I don't hire trainees. What I really am doing right now is hiring into the facilities and in the serology and hiring when we're doing that, working with Anne-Claude to hire into her team. And these they're mostly technicians. They could be research associates, people who have PhD level, data analysts, facility managers. So really people who have those supporting roles in the academic.
Right. So you're looking for really like those experts in those fields to really get the facility going rather than someone who will be coming in with a different background and like putting the time to learn an entirely new thing, correct?
Well, it depends which level we're hiring at. When we are hiring in for entry level, like we'll hire in, say, maybe what we would call a technician 1 or a technician 2. And these are people that would be coming out either just fresh out of a bachelor's or maybe with 1 to 2 years experience for a master's. And if they're coming into the proteomics facility, they don't necessarily have had to work with a mass spec before. They might have a degree in biochemistry or a degree in molecular biology, and maybe they've done some work through a co-op or 1 year doing protein purification. So we do hire at those levels, and then people do grow in those roles and get promoted up. And sometimes our facility managers have been working in the role. And like we just had a facility manager leave recently for proteomics, and we had 2 senior people under, and we promoted them up to become a scientific and operations manager. So there are ways to get promoted within. I just
Yeah. So
biggest mistake. What is the biggest mistake people make when they're interviewing for a job? I like to see that people have come prepared to the interview. We have a website, and it's amazing. Sometimes people come in and they haven't looked at the website. They don't know what we do. So I think that is one kind, because you're looking for people that come in and want to learn and want to show that they can do some background working and kind of go above and beyond. So I think that is one mistake that people do. They don't seem to become prepared for it. I like people who think about what questions to ask. We have people that ask, what do I need to be successful in this role? Like, what would you expect from me from day to day? Just those kind of things where, they've thought about the role and how they can fit into it, and they're looking to make sure it's a good fit for them as well. So I think those are 2 key things. I like people to be as kind of truthful as I can. Like when I was in the company, we were trained in how to interview people. And they said, like, what you want to do is you want to present yourself as the same person that interviews as who they'll get day to day as their boss. At the same time, I think as an interviewer, it's good to make sure that you present yourself in a way that fits with how you will continue on in the role you're hired. So I think those are kind of the things I can think of.
what's usually the interview process like for the lab that you're in? Because I know it could, depending on the lab, it could be very, very informal or like super formal, where you have multiple rounds of interviews with different people in the lab.
Yeah, and you know, some of that could depend on the level, but we usually have and we don't have any pre-screening, but like for me, others lab, but we don't pre-screen by HR. Who's ever hiring, like if it's me and a facility manager or me and like a senior person, we go through resumes ourselves. I just feel better that way. I'm sure HR could do it, but sometimes you can pick out something that HR may miss. Like maybe you've asked for a degree in molecular biology and somebody has something in physiology, but they've done this super cool, I don't know, internship or something, or just whatever. So I like pre-screening all of them. And then we have a first round of interviews. And we do this with at least 2 people, and that's actually recommended by human resources, just to make sure. I don't know, it's hard to say. I mean, there's certain things you can ask in interviews and there's certain things you can't, and there's certain things you always worry about. So having a couple people, I think, is important on the side, and you get different perspectives. So I think there's value there. And we usually have a predefined set of questions, and we ask these of all interviews, and we take notes so that when we're done the first round of interviews, we can be as systematic and fair as possible. And I should say that when we have the CVs, before I even looked at one CVs, I have a rating scheme. Like if I've asked in it that you must have a degree in such and such, you must have this experience, you should have this, I rank all those qualifications, and then I rank all the CVs so that people get up to the top. And I think that's important because there are biases raised, even unconscious bias that we know when you interview. And I think I try to do all these to have as fair a process as possible. And then we have just now, starting back with real person interviews, we have about 2 to 3 people that ask all those questions. And if it's an interview in person, then we'll give a tour so they know where they'll be working. And then often we'll call them back. Like, so then we'll, select from there about probably, I mean, it depends on the interview, but up to 2 or 3 candidates. We'll call them back for another one. And then they'll meet more team. Like in serology, when we're hiring, we already had like 8 people. They would meet like with each of the people on the team. They would go through like more of what would be expected if they hadn't met with, in this case, Aunt Clovis doing the hiring. If they hadn't met with her, they would meet with her. So that's the 2 rounds that we usually do. And sometimes these take a long time. We take the time to talk to like the preferred candidates, so there's not that many surprises if they on either side when they show up.
It's like a month process or like a few weeks?
No, because when you do hiring, you usually want to hire someone sooner. And you want to make sure that if they're looking, that they don't find another position. So, if you have the ad out for 3 weeks, then, usually within 2 weeks, you want to be contacting them and then doing the interviews within a week or so after that. So you're trying to make sure from the time the job closes as much as you can, and time slide because people get busy, that within a month you found a candidate to make an offer from.
So I was just curious, have you gotten like CVs from applicants that have like gaps in their resume, and how would you generally deal with those?
Yeah, so I mean, there are things that you do kind of look for, and even if it's just to ask a question of a promising candidate, sometimes you look at gaps, and then sometimes you also look at people who had really short stints in their employment, and at the time you can ask them to kind of walk you through the CV. And I mean, it's certainly recognized that there are times when people take breaks in their career. I mean, certainly as someone who talked a lot through this one of work-life balance, that that is a consideration, that people might have taken time off for different reasons. And sometimes you find like super great candidates that have had time off and they're really motivated to get back in and show what they can do after they've had those breaks and gaps.
Right.
And I think that's more and more, even at senior levels, even at the PI level, where, you're encouraged to explain if you have a break in your career, why you had it, so you're not judged negatively
Right.
because of it.
That's true. We have a question in the chat. Do you think that COVID-related research needs are decreasing, and are they shifting to now a different problem?
Yes. So, we're still doing some work with COVID-19. Some of the questions are now is like, we now have a highly vaccinated population, and also many of them have undergone multiple infections. And, how does that change? Does it increase longevity or the quality of the antibodies? And what's also interesting, because we have some people, very few, who never got vaccinated, never got infected. They're at the point, maybe 1% now in Canada. All the way up to people who probably had like 5 booster doses. So we have a very wide range. And in one of the meetings I've had along the way, it was with someone from public health, and they looked at, a chart of kind of, people in different categories and antibody levels. And he goes, that's it. And they honed right in the people with low antibody levels. And they're like, these are the people that are going to fill up my hospitals. So I think it's still kind of good to know the vulnerability in the population. But of course, we're learning, we know a lot more. So where things are starting to shift now, and there's actually a call out by the government for pandemic preparedness, for people who will start looking to see like what might be the next pandemic and how do we better prepare Canada for it, including our biomanufacturing. Because, that really did come up a bit short in the last one, like all our vaccines came in from the US. So we're shifting too, like we built up this infrastructure in the teams, and now we're working with colleagues to think, can we build out assays for influenza, including the avian flu that is a concern for our agriculture, for our wild birds, and whether it can cross over to humans. And then, what's the next thing? There's we had a flare up of monkeypox through this. There's the annual influenza. So we are looking at how we can take what we've done and what we've learned and, shift it to like greater surveillance across respiratory viruses, and also be ready for that next pandemic.
Is there also like a focus on doing more longitudinal studies on like effects of COVID at this point?
So there are still a lot of studies and funding on, what's called different terms, including long COVID. We have worked with some projects that have been doing this. So, I think I don't know how much of that will be what we do, measuring like antibody levels going forward, or just kind of measuring and just understanding like what makes people get this, so there's a lot of studies of those people's like immune system, sequencing, transcriptome, etc. And looking at the long-term effects. So those studies are still ongoing. I don't know how much we'll be measuring antibodies for them, but if those teams want to continue doing it, we'll be there to do the testing.
And then the last question I really had was actually to do with the NBCC facility. What kind of, because I assume it's like a facility that it's open to all the labs that are at in the institute. What kind of work is generally done on those labs, and is there room for training for people that are not part of the institute to come in and carry out some of their projects in there?
Yeah, so first of all, and maybe I didn't say this clearly enough, the NBCC is available and accessible to scientists across Canada and even internationally. So if anyone's interested in our work, they can just contact me, or if you go to the website, you can get all the contact information. So we certainly do work with people from across Canada and the Institute. We get a lot of people working like predominantly health related. I think we just starting to work with some people interested in agriculture, but we are, we work in the hospital that's at our institute works on. So a lot of people involved in understanding cancer signaling pathways. They may do like a CRISPR screen looking for an essential gene or vulnerability in cancer. We did work with Daniel Droche's lab, for instance, where they found that in by CRISPR screen in cyclin E amplified cancers, that the kinase PKMYT1 becomes essential, and they started working with Repair Therapeutics, and they've got an inhibitor that just went through phase 1 clinical trials. But while they were doing all that, we helped them with sequencing of their CRISPR screen. We helped them with imaging as they were validating their initial targets. We can do mapping out protein-protein interactions for those kind of things. So that's kind of the value of the center. And yeah, definitely, if anyone's interested, they can just contact me.
Okay, awesome. I don't think we have any other questions in the chat. So thank you so much for joining us and sharing your experience with us.